This video is a recording of a presentation held at 12 August 2021 on the conference of the International Society of Seed Science. The topic of this presentation is a proposed new seed quality strategy to obtain more resilient cropping systems. The strategy has been developed in the frame of the European LIVESEED project with financial support of the European Commission. Dear participants, I'd like to discuss with you a strategy to provide farmers with high-quality seeds for resilient crop production. I thank the organizers for allowing me to do this in an oral presentation. The last 4 years, my team has been involved in a European project called LIVESEED to aid in increasing the availability of more adapted varieties and organic produced seeds for organic agriculture. One of the topics was the development of an organic seed quality and health strategy. And I present the proposed strategy also on behalf of my colleague Jan Kodde and LIVESEED partners Stephanie Kletke and Frederick Ray. The European Green Deal is a strategy of the European Union for sustainable development. One of its aims is to increase the agricultural area on an organic practice to 25% in 2030 and to make conventional food production also more sustainable. Seed quality is an important aspect in crop production. A new strategy is needed because several challenges remain on health and quality of organic seeds. It should be noted that with conventional seeds, seed health issues will also increase, as chemical crop protection measures are increasingly banned. Also in this sense, the strategy may as well hold for conventional farmers. The strategy needs to be in accordance with the organic agricultural principles on health, ecology, fairness and care. Our proposed strategy is partly based on our own research data, but mainly on recent scientific findings. There is large diversity in seeds. Most publications and also the lectures on this conference consider the seeds they use for experiments as representatives of a species, for instance lettuce or Arabidopsis. In my experience, specific qualities of a seed batch used for experiments should also be taken into account. There is variation between varieties of a species and between different seed lots. The history of a seed batch, including seed production and storage, has an effect on its physiological quality, on the vigor and field performance. Moreover, as plants and animals, seeds are not sterile. They contain a large variety of microorganisms, and together they form a so-called holobiont. In the past, the attention to the microorganisms and in and on the seeds was directed at pathogens, but now we know that seeds contain many other microorganisms. Some can even be beneficial and counteract pathogens. Germination tests on filter paper and optimal conditions in the lab can show a good germination, but in the field, the germinating seed and seedlings face multiple stresses. It can be too wet, too dry, too cold, too hot. Pathogens can be in the soil, and all these can result in lack of seedling and crop establishment. We mimicked one of these field stresses in the lab by adding spores of a fungal pathogen called Alternaria rudicina. These spores were added to germinating carrot seeds in the petri dish. And we observed that seeds with a lower vigor, that was induced by a few days storage at high humidity, that they are much more sensitive to the pathogen compared to the high vigor seeds. This clearly indicates that seed vigor has an effect on the sensitivity to pathogens. Seed vigor is therefore an important aspect in seedling establishment. Low vigor seeds have more difficulties to cope with field stresses. High vigor seeds are needed for rapid seedling emergence and crop establishment. The seed microbiome adds a whole new dimension to seed physiology. It's a relatively new field of research, but can have implications for our experiments. The research group headed by Professor Gabriele Berg at the University of Graz, Austria, is one of the leading teams in this field. Here 2 publications in which your team was involved, showing the potential of the seed microbiome for seed quality and seed health. Microbiomes within plants or seeds are called endophytes. And the publication on the left showed that microbes present in the soil enter the tomato plant and subsequently enter the seed and are transmitted as endophytes to the next generation. And it appears that microbes beneficial for the next generation are preferentially transmitted. And this is likely the result of millions of years of co-evolution of plants with their microbiome. A second publication from this year that showed that resistance of rice seedlings to a bacterial disease is not based on plant genetics, but on the seed microbiome. Comparing 3 different rice seed productions with the same cultivar showed transmission of the resistance-inducing bacteria with only 2 of the 3 seed productions, clearly indicating a seed production effect. Seed disinfection treatments to get rid of pathogens will also remove part of the seed microbiome. With developing seed disinfection protocols, not only the effect on seed vigour should be taken into account, but also the effects on beneficial microorganisms in the seed microbiome. Interestingly, physical disinfected seeds seem to have more frequent problems with pathogens in the soil from conventional farms compared to soils from organic farms. The latter may be related to a more biodiverse soil microbiome in organic farming managed soils. In a proposed strategy for development of more resilient cropping systems, we suggest to place more emphasis on seed vigour, because early emergence in the field has a strong effect on crop establishment. The handbook of the International Seed Testing Association lists only a few vigour tests, including the so-called controlled deterioration test, and there is a clear need for more vigour tests. The seed microbiome and its diversity can be used as a natural mechanism, aiding the seeds and seedlings in its tolerance towards biotic and abiotic stresses. Both aspects should be taken into account during seed production, harvesting and processing, and eventually also in seed quality testing. The research performed and published thus far is very promising, but sure, more multidisciplinary research is needed. This proposed strategy was developed in the frame of the European LIVESEED project. That project is supported by the European Commission and the Swiss government. My team also received support from the top sector Horticulture and Propagation Material, and I want to acknowledge also the other parties in the LIVESEED project for their contribution and discussions on the strategy. With this, I end my presentation and invite you to raise questions and to discuss on the proposed additional elements considering the new seed quality strategy. The objectives of the LIVESEED project are to improve transparency and competitiveness of the organic seed and breeding sector, encouraging greater use of organic seeds and to increase supply of improved varieties and high-quality seeds. If you're interested in the project and its outcome, visit the project website at www.liveseed.eu. You may also contact the authors of this presentation for more information or collaboration.